Sorry.
Good, visible?
Yep.
See.
Yes.
Excellent. Page are we going to?
Well, that would be 227. 2027.
Mm, 200.
Oh. Well, I tried.
So
If only the book went up to that high. Cindy, you're unmuted.
So then, yeah, just the same thing, Jaime, just the page before with the.
yeah, 226.
Yeah.
Okay. And then you guys again.
Good stuff.
Share screen on the.
I think this week is the longest chapter we will have read.
Oh, really?
I think so. I think usually they're like 8 to 9 pages. This week is like
Yeah.
12. That's okay.
Here we go.
Are you sure you can't hear kids screaming in my background?
In
Okay.
your basement? Excuse me?
In my background.
Just kidding.
They're so loud. There's like a pool like behind in my alleyway, and there's kids like having a great summertime fun.
School's out. Everybody's partying.
I know.
Should we start letting people in? We don't have that many people in the waiting room.
I'm wondering, should we wait just one more second to see if Lauren?
Sure.
I can text her too and ask her.
I mean, she knows
Okay.
it's happening, right?
Well, there was a debate whether or not she's flying to Mexico today or something.
Mexico.
I thought Lily said that she was going to be added to going to Mexico this week, or this weekend, but I don't know when. Or maybe it's all false information. Could be my dad's going senile and hearing things.
So this chapter is about mushroom sex.
Kinda, yeah. There's a big part about mushroom reproduction,
And cloning.
and it's
Other spores and yeah, a lot of good
yeah. Well, when we were doing it on Tuesday, the review, Cindy and Jen were pretty confused about some of the
stuff.
stuff. So
The
we
birds and the bees kind of stuff.
Yeah,
yeah, because
I had never heard the birds and the bees from mushrooms. It's really complicated.
yeah.
My mother never sent me down for that one.
So yeah, it's about the traditional way, and then also there's like a very weird way that Matsutake has sex with itself.
Your drawing said they can have up to 4,000 different sexes.
4 to 10,000.
To 10,000, 4 to
Yeah.
10,000.
So most of them have like 4 or 8, but there's one Schizophilum commune split gill. You'll find it everywhere. But that one has 10,000 different ones.
Well,
Yeah.
Okay, Lauren said she's joining.
right.
Can you hear the airplane?
Oh, a little bit. Do you have your Zoom settings set to like the choir? Okay.
I fixed,
No.
but it still picks up everything.
Oh, okay.
Olive, don't breathe so loud.
Lauren's here.
Cool.
So should I let the rest of us in?
Hello.
Hi. How are you?
Oh, you're
I think
muted.
you're mute.
I'm good. What an exciting day, reaching the primordial floodplain and all that clay. I've been waiting for that moment for a long, long time. So it was really gratifying to see it.
It's pretty cool.
Yeah.
Bless you.
God bless you.
Thank you. Are we ready to open up the room?
Absolutely.
Yep.
Hello, everyone, and welcome to Learning and Mending. As we are all starting to sign in and get situated, I will start with our embroidery pattern of the week. And this week I chose a mule fat design based on, I believe somebody last week said that they felt they were never going to get through their mule fat design. So I'm offering you some hope. They can be completed. This one Izzy made, and it's absolutely beautiful. I have to tell you, this part up here, it just feels so 3D and just like it's actually growing. And so when I received this, I thought this is perfect for this week. So whoever it was last week that said that they had the mule fat and it seems like it's forever, it's possible. So continue on your journey of your mule fat embroidery. And I will start with introductions. I'm Jen, and I'm calling in from Studio City, and I use she, her pronouns. And then what we'll do for the next couple of minutes is we'll just pass it around the room. So I will pass it to Cascade.
Hi, everyone. Happy to see you. I'm Cascade, she, her pronouns, calling from Jefferson Park, working on my common raven, making some progress. Thank you. And I will pass it to Cynthia.
Hi, I'm Cindy. I'm calling in from West Covina. I have a lot of background noise, but I'm working on my little bird still, and I will do the actual bird today, maybe. I will pass it on to Diego.
Hi, I'm Diego. I use he, him pronouns. I'm calling from Altadena, and I'm going to be working on a drawing today. So I'll pass it on to Mish now.
Hello, I'm Michel. I'm from Highland Park. Pronoun he, him. I will probably be also sketching logs in water. I'm not quite sure. And I will go on with James.
Hi, everyone. Yes, thank you, Mish. Hi, everyone. I'm James. He, him pronouns, calling in from downtown LA. And tonight I will be reading and explaining fungal sex later, which will be a very interesting thing. So I will pass it on to Olan.
Hello, Olan reporting in West Hollywood. I use she, her pronouns. And whoever thought they would never finish the mule fat clearly has not yet worked on the bird, which I am still working on.
Wow.
I have ventured into the bird itself, Cindy. And I will pass it on to Kavi. Is that the way you say her name?
Yes. Kavilal, calling in from Sacramento, California, he, him pronouns, and I am working on the bird. Slow start, but going through. And I'll pass it over to Susan.
Susan, I think you're muted.
God, I suddenly felt old now. I'm muted. That's a thing my kids tell me. So I'm calling in from the Santa Monica Mountains and just got my voice. Good to be here this evening. I'll pass it on to Lauren. Hi.
Hi, Susan.
Hi.
Lauren, she, they pronouns, calling in from Mexico. Working on the Common Raven. I'm glad to be here tonight. And I will pass it on to Jinda B. Or if that person doesn't want to.
Yeah, I'm Jinda,
Oh.
calling from Brooklyn, New York. Just enjoying watching the event from the last week. And want to see more. Yes.
Hi, Ansu. How about you go next?
Hi, everyone. Ansu, I use she, her pronouns. I'm calling in from Idlewild. And talk about slow, but I am making a little bit of progress on my B. Who hasn't gone yet? Should I pass it on to Jaime? Did Jaime go?
I have not gone yet, no. Hey, my name is Jaime. I go by he, him pronouns. I'm up in Lone Pine in the Owens Valley. I'll be sharing the screen today with the reading.
And I can take it from here, Jaime. Before I begin, I'll introduce my assistant, Fig. Yes, he does eat floss, and no, he does not embroider, but he likes to watch the whole process. So, with that, welcome to Metabolic Studios Learning and Mending. The Metabolic Studios Learning and Mending series is now in its 4th evolution. This time around, we are addressing disturbance ecology and what emerges in the blasted landscapes of capitalism. To do this, we will be collectively reading Anna Tsing's The Mushroom at the End of the World on the possibility of life in capitalist ruins. Growing through the last 3 gatherings, the first learning syllabus in response to ongoing racial violence, particularly at the hands of police, the second holding space for the megafires that consume the West, and the third addressing the larger reality of suffocation and the need for us to revive our breath. In order to gain a deeper understanding of these crises and how they are interconnected, we're building upon these readings in the last 3 series. Many of you have received embroidery kits from us of native plants representing the food and medicine that Metabolic Studio is cultivating. We are adding some new embroidery patterns, and we have more native plants and bird patterns available too. The theme of all the embroideries we've been working towards is biosynosis, which is an association of diverse organisms forming a closely integrated community. If you haven't received an embroidery kit, or you finished yours and would like to receive another one, please send us your name and mailing address to info@metabolicstudio.org. And Cindy will put that information in the chat bar as well. We decided to close the entrance to the Zoom space after the first 15 minutes of the reading to protect the flow and to keep a steady breath. These gatherings are being recorded through Zoom for archival purposes. We thank you for co-creating this opportunity to learn and to mend together during quarantine. Tonight, we'll read chapter 17 from The Mushroom at the End of the World by Anna Singh. This 10-week series will focus solely on this one book, and our guiding question is, what lives in the ruins we've made? Since the ongoing pandemic began, we have moved our weekly public studio practice to Zoom, where we've committed ourselves to learning and mending together. We invite you to receive these readings as a deep inhale, and we find that crafting together helps to receive these readings as a meditation. We will share a screen, and we encourage you to read a section of the text aloud with us. When there is a pause in the reading, please feel free to unmute yourself and begin where the last person left off. Silence is golden, and there's no pressure to fill the transitional spaces while we enjoy a breath and continue crafting. Feel free to jump in and continue reading at your comfort level. You don't have to read. It's also fine to enjoy listening. And now I will pass it over to James, where he'll introduce the reading and the discussion questions.
Thanks, Jen. Hello, everybody. As usual, I'll drop in and read our discussion questions for this evening. And I also just want to say that we decided that there was a part of this tonight that was a little more technical, and part of it is just maybe like a mind blowing of how fungi reproduce versus the way we know plants and animals do. So at the end of the reading, before we go into discussion groups, I'll also do like a little just short breakdown, and also feel free to ask any questions and talk about them in your discussion groups. So the first question for tonight is, the Platonic idea of species is challenged in chapter 17. For example, Chinese scientists think there are 21 different species of oyster mushrooms based on different physical differences. Other scientists say that there are only 14 species based on DNA. What are your thoughts on the definition of species? The second question is, when exploring the different possibilities on how Matsutake spread across the globe, Singh presents the idea that ecosystems move, not the fungi. Humans are one of the largest and unintentional culprits in moving ecologies, as well as other natural phenomena over huge scales of time. How does this influence your imagining of the way life moves? And with that, Jaime, do you want to pull up the reading? Cool, thank you. Reading forests. Yunnan. Identifying an evergreen oak. Oak forms interbreeding hybrid swarms. And yet distinctions are somehow maintained. Names only open the mystery. Chapter 17, flying spores. All of this, of course, all of this is, of course, speculation. Mycologist Zhangping Zhu discussing Matsutake evolution. Landscapes and landscape knowledge develop in patches. Matsutake Shiro, mycelial mats, model the process. Patches spread, mutate, merge, reject each other, and die back. The hard work and the creative, productive play of science, as well as emerging ecologies, happens in patches. But one might also sometimes wonder what moves beyond them, making them. For Matsutake, there are also flying spores. Both in forests and science, spores open our imaginations to another cosmopolitan topology. Spores take off towards unknown destinations, made across types, and, at least occasionally, give rise to new organisms, a beginning for new kinds. Spores are hard to pin down. That is their grace. In thinking about landscapes, spores guide us to in-population heterogeneity. In thinking about science, spores model open-ended communication and excess, the pleasures of speculation. Why spores? Koji Iwase first started me thinking about spores. We were having lunch in Kyoto with Shiho Satsuka and Michael Hathaway. The tape recorder wasn't on. I was curious about why Matsutake is so cosmopolitan. How did it spread around the Northern Hemisphere? Dr. Iwase is generous with foreigners and willing to guide them. So he mentioned that the stratosphere is full of fungal spores. At those high altitudes, they blow around the Earth. It's not clear, he continued, how many of these spores survive to germinate in distant places. Ultraviolet radiation kills, and most spores are viable only for a short time, perhaps a few weeks. He didn't know if a Matsutake spore could survive to germinate on another continent. Even if it did, he explained, it would have to find another germinating spore. Without fusing, it would die in a few days. Still, over the course of 1 million years, one might imagine that spores could spread the species. There is something about the stratosphere that inspires airy dreams. Imagine spores circling the globe. My thoughts took off with drifting spores, chasing my protagonists across eons, across continents. I took my questions to mycologists here and there around the world, chasing their thoughts too, through the stratosphere. I found a cosmopolitan science of speculation about origins and the making of kinds across space and time. Unlike the discontinuous patches of applied forestry, the science of Matsutake speciation is not patch-like. There are strong winds of international consensus about methods. The materials, mushroom samples, and DNA sequences circulate across borders. Individuals and sometimes labs develop stories, bits of expertise, and even biases. But there are no schools, no patches. All this work is off the clock. No one gives out grants to study the eon crossing travels of a mushroom. Scientists turn to these questions out of love, and because the methods and materials are there. Perhaps one day the combined results and speculations will lead us, like spores, to something new, they reason. For now, it is just the pleasure of thinking, the spore-filled, airy stratosphere of the mind. But what are these materials and methods that circulate? Henning Knudsen showed me the University of Copenhagen Botanical Gardens fungal collection, of which he is curator. Type specimens are stored here, drawers and drawers of folded envelopes, each guarding a dried fungus. When a new species is named, the namer sends a sample to the herbarium, and those specimens become the type for that species. Researchers from around the world can ask to see the type. The herbarium sends the original material. The herbarium system emerged with the Northern European passion for identifying plants, which also resulted in Latin binomial names. It was a feature of European conquest. It also created the basis for transnational communication through the circulation of specimens. Researchers around the world know species through type specimens collected in herbaria.
Dr. Knudsen does not think Matsutake spread through spores in the stratosphere. It's just too improbable that they would find mates. Instead, their distribution followed the forests. They spread together with the trees. This took a long time, but across the northern half of the earth, many species spread ever so slowly together. Some, such as Boletus edulis, may have spread across the top from Alaska to Siberia, but the homogeneity of northern species is overstated. Many species that used to be seen as uniformly found across the global north turn out to be different species, he said. The rejection of uniform cosmopolitan species draws not from the circulation of herbarium samples, but from a revolutionary new technology, DNA sequencing, which offers a new way to define, quote, species. Mycologists examine particular DNA sequences, for example, the internal transcribed spacer, ITS region, that tend to be conserved within species but show variations across them. Jean-Marc Moncalvo, Dr. Knudsen's counterpart at the Royal Ontario Museum in Toronto, explained that more than a 5% divergence in the ITS sequence indicates a new species. DNA sequencing does not reject the materials and methods of herbaria. Most comparisons across species use herbarium samples, but there's a new material here in circulation. The DNA sequences themselves. Databases have made it possible for scientists around the world to consult DNA sequence by others. The simple precision of DNA sequencing has taken the scientific world by storm. There are no alternatives. It seems so powerful that scientists keep making up questions based on the availability of this answer. Of course, there are still pockets of difference. Dr. Moncalvo explained that as recently as the 1980s, Chinese mycologists had trouble communicating freely with Europeans and North Americans. One Chinese mycologist sent him samples of fungi hidden between the pages of reprints. As a result of isolation, he said, Chinese taxonomies are strange. Internationally, there are no rules for naming a genus, the first name in a Latin binomial. So Chinese taxonomers have added China to genus names, assembling Sino-Boletus instead of Boletus and confusing foreign counterparts. Furthermore, they recognize species indiscriminately. They claim to have 21 species of oyster mushrooms in Yunnan, but there are only 14 species recognized in the world. Tiny morphological differences are given too much attention, but this is changing now, he said, as young scientists with international training take over. What do these materials and methods tell us about, quote, kinds?
Species has always been a slippery concept, and DNA sequencing, despite its precision, has not made it easier to handle. Classically, species boundaries were defined by the inability of individuals on each side to mate and produce fertile offspring. That's easy enough to figure for horses and donkeys. They mate but do not produce fertile offspring. But what about fungi? Dr. Moncalvo walks me through what it would take to find out if two different fungal strains were species, according to this definition. You would need to germinate one single spore of each in culture, get those spores to mate, somehow force them to produce a mushroom, then get its spores to mate and produce mushrooms. For a fungus such as Matsutake, for which no one has succeeded in producing a single mushroom in culture, and whose spores don't even germinate if alone, such experiments are hardly worth conceiving. Besides, thank you. Besides, Dr. Moncalvo added, imagine the hapless graduate student who devoted a dissertation to finding a species boundary of even the easiest to handle mushroom. Where would he or she get a job? All this matters in getting to know Matsutake across its diasporic locations. 20 years ago, there were many, many species of Matsutake scattered around the Northern Hemisphere, with more emerging constantly as scientists found them. Now there are just a few, and growing fewer. This is not because of extinction. DNA sequencing in the ITS region has allowed scientists to argue that most of those kinds of Matsutake are really just one kind, Tricoloma matsutake. T. matsutake now appears to spread across most of the Northern Hemisphere, not just across Eurasia, but into North and Central America. Only Tricoloma magnivillare, the matsutake of the Northern American Pacific Northwest, is continuing to stand clearly as a separate species, and even it very close in its DNA signature to T. matsutake. The precision of DNA sequencing, which allows such determinations, also undermines confidence in the species as the basic category for understanding kinds. I first met Kazuo Suzuki, now president of Japan's Forestry and Forest Products Research Institute, when new results were coming in about the identity of China's oak-loving matsutake, at that time called Tricoloma zhangjiai. In Japan, matsutake are associated with pines. Only false matsutake are found with broadleaves. The association between matsutake and conifers seemed part of its species definition. DNA studies showing the close relation between China's oak-loving matsutake and Japan's exclusively pine-loving ones caught researchers by surprise. Dr. Suzuki brought his younger colleague from Tokyo University, Dr. Matsushita, to our meeting to tell me the news himself. His examination of the ITS sequence had shown no species difference between oak and pine lovers. But Dr. Suzuki, who had worked with matsutake for many years, did not accept this finding as the whole story. It depends on what question you ask, he explained. He told me about Armillaria root rot, a complex of species in which clear species boundaries may not be relevant. Armillaria root rot spreads across whole forests, stimulating boasts of the largest organism in the world. Differentiating individuals becomes difficult, as these individuals contain many genetic signatures, helping the fungus adapt to new environmental situations. Species are open-ended when even individuals are so molten, so long-lived, and so unwilling to draw lines of reproductive isolation. Armillaria root rot is 50 species in one species, he said. It depends on what you are dividing species for.
I remember the discussion vividly. I was at the edge of my seat. Dr. Suzuki was treating species in the way cultural anthropologists treat their units, as frames that may be continually questioned to retain their use. the kinds we know, he implied, develop at that fragile junction between knowledge making and the world. Kinds are always in process because we study them in new ways. This makes them no less real, even as they seem more fluid and beckoning of questions. Ignacio Chapela, a forest pathologist at the University of California, Berkeley, was even more adamant that the idea of species limits the stories we can tell about kinds. This binomial system of naming things is kind of quaint, but it is a complete artifact, he told me. You define things with 2 words, and they become an archetypal species. In fungi, we have no idea what a species is. No idea. A species is a group of organisms that potentially can exchange genetic material, have sex. That applies to organisms that reproduce sexually. So already in plants, where out of a clone you can have change as time goes by. You have problems with these species. You move out of the vertebrates to the cnidarians, corals and worms, and the exchange of DNA. And the way groups are made are very different from us. You go to fungi or bacteria, and the systems are completely different, completely crazy by our standards. A long-lived clone can all of a sudden go sexual. You can have hybridization in which whole big chunks of chromosomes are brought in. You have polyploidization or duplication of chromosomes, where a completely new thing comes out. You have symbiotization, the capture of, say, a bacterium that allows you to either use the whole bacterium as part of yourself or use parts of the bacterium's DNA for your own genome. You've become something entirely different. Where do you break down the species? To compare different kinds of Matsutake, Dr. Chapela used herbarium species, as well as fresh samples and sequenced ITS region DNA. But he refused to imagine his results as fixed species. You start getting these groupings that you can only name relative to each other. You can't call them a species. In the old taxonomic approach, you say, this is my ideal. It's completely platonic, and everything is going to compare as a missed approximation to that ideal. Nobody will be the same as this. But you compare and you see how close they are to this ideal. If it becomes too different by whatever measure, the measures are completely arbitrary. You say, oh, this must be a different species. To avoid a false scientific cover, he speaks of Matsutakes as all the varied kinds that enter the Japanese trade. His study did, however, find distinct genetic groupings by region. That means, he said, that genetic materials are not freely exchanged across those regions. If you see good patterning, if you see good separation, that tells you that there is not much exchange between these groups. These data show that cross-regional exchange of spores is unlikely on a regular basis.
One down for the long distance travel of spores. but other possibilities have just become more thrilling. How then do kinds travel? Dr. Chapella, working with his colleague, Dr. Gabelotto, has a story to tell about Matsutake's travel. The Eocene ancestral population, he argues, developed in North America's Pacific Northwest, where T. magnivelare continues to associate with both broadleafs and conifers in resonance with that broadleaf-loving ancestor. The rest of the Matsutake group jumped to conifers and has followed conifer forests ever after around the Northern Hemisphere. Where conifers retreated into refugia, Matsutake followed, especially with pine. Wherever the pine forest went, Matsutake went too. Migrating across the Bering Strait, Matsutake colonized Asia and then Europe. The Mediterranean Sea blocked gene exchange between southern Europe and North Africa. Populations on each side are independent extensions of the vast Eurasian trek. Meanwhile, Chapella and Gabelotto imagined that southeastern North America was colonized by Matsutake from the rich pine oak refugia in Mexico. Their story was shocking in part because at the time they published, most people thought of Matsutake as an Asian species complex. After all, only Japanese and Koreans loved Matsutake and thought of it as their own. How could it be a North American mushroom that came late to Asia, even if millions of years ago? Chapella and Gabelotto date the separation of T. magnivelare and other Matsutake as having occurred 28 million years ago with the rise of the Rocky Mountains. Indeed, not everyone agrees with the story they tell. This is an open-ended field. Dr. Yamanaka of the Kyoto Mycological Institute argues for a Himalayan origin for the Matsutake. Many new species came into being with the rise of the Himalayas, which forcefully threw old kinds into new environments, stimulating difference. At the time of Chapella and Gabelotto's research, the evidence of host differentiation among Matsutake in southwest China was not readily available, at least in California. It turns out that Chinese Matsutake associate not just with conifers, but with quercus, as well as Castanopsis and Lithocarpus, which find their centers of species diversity in the Himalayas. Dr. Yamanaka reminds me that the major broadleaf host of North America's T. magnivelare is tan oak, the only non-Asian Lithocarpus. Might this be a clue? Dr. Yamanaka found Matsutake shiro in China, associating with both conifer and broadleaf hosts. He argues for Himalayan origins based on, in part, on the sheer variety of mycorrhizal arrangement in that area. Diversity is often a sign of time and place. Yet even new research has shown that Southwest China's Matsutake are not particularly genetically diverse, at least in the ITS region most commonly sequenced by researchers. They are a whole lot less diverse than Japanese Matsutake, which everyone agrees to be latecomers on the evolutionary scene. But this does not mean they are a newer population. Xiangping Xu of Canada's McMaster University suggests that Chinese Matsutake just fill up more of the available space than in Japan. This saturation, he points out, can lead to longer living clones with less genetic competition. The stress of industrial pollution might also lead to genetic competition in Japan. Southwest China is far less industrialized. Diversity is not just about time and place.
Dr. Xu brings back the questions of spores. Many mushroom species are widespread. They are opportunistic. Wherever there is food, they can survive. Dispersal is not such a significant barrier for most of them. Here he brings up the panspermia hypothesis, which posits that spores are everywhere, traveling even in outer space. For most microbial species, you can find them everywhere. Dispersal is not the barrier. It's whether they're able to survive in those environments. He jokes. It's like Chinese now. They're everywhere. If there are business opportunities, you're probably going to find Chinese. If there's a small town, you'll probably find a Chinese restaurant. We laugh together. He talks of how well spores are dispersed. For many species, there are limited genetic differences among populations from very different geographic areas. One example is the bacteria in our mouths. He says that the bacteria in the mouths of middle-class urban Chinese are vastly different from those of their peasant neighbors, but just the same as the bacteria of North Americans on a similar diet. It is the environment, not the location, that matters. For many fungi too, he confirms, dispersal is not the problem, especially since humans emerged. There's a new thought. Humans? Dr. Zhu is not the only one who thinks that human trade and travel have dispersed fungal spores. Dr. Moncalvo finds that very significant, although he disagrees with the idea that spore clouds are everywhere. Mushroom populations are restricted and well-defined. The same morphology on two different continents is usually separated by genetic distance. There is exchange through spores, he argues, but it is occasional, not constant. But exchange may be more common now because there is more trade and more travel. For example, Amanita muscaria was transferred to New Zealand in the 1950s and is now spreading. It is not even out of the question that Matsutake spread across the Atlantic with human contact. There are a lot of Scots pines here. Scots pine is a major North Eurasian Matsutake host, but it is not native to the New World. Canadians, they still have the queen on the coin, right? So they think the pine seedlings are coming from Her Majesty's garden. Excuse me. So they think the pine seedlings that are coming from Her Majesty's garden must be better quality than native pine. He shakes his head in mock horror, but it is a serious point. Perhaps Matsutake traveled to eastern Canada on the roots of pine seedlings. Dr. Moncalvo does not dismiss the possibility of spread without humans, but he does think the spread must be recent because Eastern North American Matsutake are so very similar to Eurasian ones. And he adds, shocking me, who knows which way the spread went, especially if we find the two species, Western America's T. magnivelare and cosmopolitan T. matsutake, coexisting in Central America and possibly on the southern Appalachians. That might be the origin. One, T. magnivelare, has been stuck on the West Coast. The other, T. matsutake, has moved on. That is something a phylogenetic study should be able to tell. How might both species have come to Mexico, I asked. It was a southern refugia during glaciation, he explains. It's a well-known phenomenon. The southern limit of oaks and pines are the mountains in Central America. You don't find them in South America. And you find them with altitude. When it gets cold, everything moves south. When it gets warm again, they move up to higher altitude. 3,000 meters in Mexico is like sea level here. This can also explain some shuffling. Populations will grow back from local refugia, but they are not salmon swimming back to the stream in which they were born. There is no reason that one goes one way or the other way. It is the ecosystems that moves. It's not the fungus that moves. It's the ecosystem that moves. No wonder humans move so many other species without meaning to. We create new ecosystems all the time. And it's not just humans that change things. I rather think it can sometimes be events, Dr. Moncalvo explains, to my repeated questions on how kind spread. That's something that many people cannot grasp. The time frame is huge. The tectonic separation between the southern and the northern hemisphere is 100 million years. So we find different species in the southern hemisphere and the northern hemisphere. Australia is a great example. So people say, oh, they separated 100 million years ago, but it's not true. Now we have molecular data. We see it's incorrect in most cases. They are isolated, but there is sometimes transfer. But the transfer is not all the time. So we don't have something homogeneous. There could be one transfer per million years or per 10 million years. The transfer could be anything. It could be a tsunami wave starting from the Philippines and crossing the equator. They usually don't cross the equator, but in 100 million years. And carried on the top of the wave, some soil and some wood with some animals hanging. Could also be wind. It could be anything. Once mycologists thought that southern and northern hemisphere mushrooms have been isolated for 100 million years, but DNA sequences now show that this could not possibly be true. For Amanita, for example, there are many groups with north-south ties, rather than just a single hemispheric dichotomy. Assumptions about slow and constant mutations in place are being displaced by attention to unusual events, indeterminate encounters.
How do kinds emerge then in local populations? Dr. Zhu explains it. Scale matters. One cannot use the same tools to study cross-continental and local diversity. The ITS region of fungal DNA is fine for studying big blocks of regional difference, but it's useless to study local populations. There, a completely different clump of DNA is needed to judge the variations that separate one group from another. Dr. Zhu has found that single nucleotide polymorphisms, SNP, are good for population-level differentiations. With this tool, he studied Matsutake populations in China, finding little genetic difference between oak and pine-loving Matsutake, but a significant geographic separation across sampled regions. Most important, perhaps, this separation added evidence that sexual reproduction is important in Matsutake populations. Spores rise again. In the world of fungi, this is not at all self-evident. Fungi propagate through many mechanisms, and sexual reproduction through the Matsutake mating. Through the mating of germinated spores is just one. A good deal of fungal propagation is clonal. Some clones, including those of the famous Armillaria root rot, are large and very, very old. Fungi also propagate through asexual spores, which are produced in times of stress. With their thick walls, they withstand hard times to germinate when better conditions return. For some species, sexual reproduction is absent or rare. For Matsutake, however, the evidence suggests that sexual spores are important. This isn't investigated by examining the genetic composition of clonal patches? Are they mutating independently or exchanging genetic materials? For example, do you find more genetic diversity in older forests rather than in younger ones, where you would expect a founder effect rather than free spore dispersal? For Matsutake, the answer to this last question is yes. Spores appear to be exchanged among patches of mycelial growth. However, landscape features can block the exchange of spores. Researchers found that ridges, for example, block genetic exchange among Matsutake populations. This seems familiar enough, but don't relax. Matsutake does something strange and wonderful that can turn your idea of sexual reproduction upside down. It was another meal, tea this time, in Tsukuba City with Hitoshi Murata of the Forestry and Forest Products Research Institute and Matsutake World's team member, Lieba Vair. I was so excited when I understood that I spilled tea all over my tray. Dr. Murata had been studying the genetics of Matsutake populations. It was a painstaking process since Matsutake is not an easy research subject. Figuring out how to get spores to germinate was itself a problem. They germinated, he found, in the presence of other Matsutake parts. For example, mushroom gills. This suggested that spores might germinate best on living shiros, that is, mycelial mats, including that of the parental body that gave rise to the mushroom. And what happened then when they germinated? This is where his research revealed something wondrous. Matsutake spores are haploid, that is, bearing only one series of chromosomes rather than paired sets. We might expect them to mate with other haploid spores, thus making full pairs. They do. Human eggs and sperm join that way, but Matsutake spores are capable of something else. They can join with body cells that already have chromosomal pairs. This is called daimon. Mating from the prefix of 2, the number of chromosome copies in the fungal body cells, and 1, the number in the germinating spore. It's as if I decided to mate with, not clone, my own arm. How queer. The spore brings new genetic material into the shiro, even if it is the shiro's offspring. But the shiro itself is a mosaic, a combination of multiple genomes. Even emerging from the same shiro, different mushrooms might have different genomes. Even emerging on the same mushroom, different spores might have different genomes. The genetic apparatus of the fungus is open-ended, able to add new material. This adds to its ability to adapt to environmental shifts and to mend internal damage. Evolution in one body, the fungus can discard less competitive genomes to pick up others. Diversity emerges right there inside the patch.
Dr. Murata explains that he was able to ask these questions because of his unusual background for a mycologist. He was originally trained in bacteriology. Most mycologists come from botany, where they see one organism at a time, or ecology, where they see interactions across organisms. But bacteria are too small to care about one at a time. We know them in patterns and masses. As a bacteriologist, he knew about quorum sensing, the ability of each bacterium to chemically sense the presence of others and to behave differently en masse. From his very first studies of fungi, he found quorum sensing there. In fungal mosaics, each cell line can sense the others, forming mushrooms in unison. By examining fungi differently, a new object came into sight, the genetically diverse fungal body, the mosaic. Mushrooms with genetically diverse spores, mosaic bodies, chemical sensing that creates communal effects. How strange and wonderful the world. I struggle. Isn't it time to come back to patches, incompatible scales, and the importance of history? Shouldn't I return to multiple rhythms, the tempos through which patches emerge in both landscape and science? But how happy it feels to fly with spores and to experience cosmopolitan excess. For the moment, the reader must make do with hasty conclusions. Spores vitalize Matsutake populations through adding new genetic materials. Mushrooms produce many, many spores, and only a few of them germinate and mate, but it is enough to keep populations cosmopolitan and diverse. Some of that diversity is within the parental bodies that produce the spores. No one fungal body lives self-contained, removed from indeterminate encounters. The fungal body emerges in historical mergings, with trees, with other living and non-living things, and with itself in other forms. Scientists speculate about open-ended questions, including the evolution and spread of Matsutake in a spore-like way. Most of those thoughts never make a difference, but the few that do can revitalize the field. Cosmopolitan knowledge develops out of historical mergings, with research subjects living and non-living, and with itself in other forms. Patches are productive, but there are also spores.
Thanks everybody for reading. I'm just going to do a quick cover of a little bit of what we just talked about with mushroom sexual reproduction, in case that was like a little abstract, but it won't take too long, so please bear with me and bear with my crude drawings. So yeah, here's the life cycle of mushrooms. Over here we have the mushroom itself dropping spores. Then the spores themselves germinate into monokaryotic mycelium, meaning they only have one half of the DNA that they need to be a whole fungus. Then two sexually compatible spores fuse together to make a dikaryotic mycelium, meaning that they have all of the DNA that they need. So here, you can't really see too well, but the little network with the green DNA and the little network with the red DNA come together and fuse, and then they have this full network with both red and green. Then that network grows, starts to form a mushroom, and then we're back in the cycle again of the mushroom drops spores. And then to show what Singh was talking about in the very unusual way that Matsutake mates with itself and prefers to mate with itself. Oh, I also forgot to highlight one of the interesting points is that fungi can have anywhere between 4 to 10,000 different sexes, just for reference. But then here is how Matsutake kind of form. So again, daimon mating, dai meaning 2 and mon meaning 1. And so this is where the mother network, the mycelium, meets with one of its own spores, and they come together. So over here, there's a full complete network with the red and green. And then you've got this spore network that only has, looks like black, but it's supposed to be blue. And then they come together and they start fusing together. So now, instead of having just one pair of nuclei, red and green, the Matsutake network could now have 3 different kinds, red and green, blue and red, and green and blue. And so that kind of speaks to how, resilient they are when adapting to environments. And also the oddness of what Singh was trying to get at is that this would be like you mating with your own arm in a strange way. So yeah. And I will just reread the discussion questions, please feel free to talk about anything else because this week was a bit heavy in many ways. But if you'd like to talk about the discussion questions, go ahead. So the first one is when exploring the different possibilities on how Matsutake spread across the globe, Singh presents the idea that ecosystems move, not the fungi. Humans are one of the largest and unintentional culprits in moving ecologies. How does this influence your imagining of the way life moves? There's that question. And the second one is, sorry, one sec. The Platonic idea of species is challenged in this chapter. For example, Chinese scientists think there are 21 different species of oyster mushrooms based on different physical differences. Other scientists say there are only 14 species based on DNA. What are your thoughts on the definition of species? Yeah, I think Izzy, you wanted to see the first one again? Sure.
Yes, please.
I'll just show it up and then we can break out.
Pretty awesome.
So again, mushroom drops spores, spores germinate, fuse together, grow out into a mycelium, and then they decide to make a mushroom and start the whole thing over again. Cool. And with that, Jen, will you send us to our breakout rooms?
I'll send you to the breakout rooms and we'll be there about 8 to 10 minutes and then we'll all come back together and discuss what we discussed individually. So I will send you now. So see you in 8 minutes.
Hey, what's happening?
Hello, welcome back.
Hey, hi everybody.
Hi, Alex.
Hi, Diego. How are you? Good to see you.
Yeah, you too. I'm good, thanks. How are you?
I'm doing great.
I think we're all back. Yep, looks like we're all back. Would anyone like to start with what they discussed in their breakout room?
I can kind of start if Izzy and other people from our group want to back up points that we talked about, which is we kind of centered on the topic of, or the definition of ecology when trying to answer the question about like humans moving ecology unintentionally and changing it. And Izzy kind of brought up the question of, you know, like how large of a definition and loose ecology can be, and how you know, where does the line, like what's not an ecology and what is an ecology in that sense? Because we were thinking, you know, from the smallest parts of your body where you have microorganisms to the larger parts of you know, the connection between geology and life. And then even if you're thinking like the lines that you start to run in are like, well, like are planets and like stars not parts of ecology, and are like subatomic particles not parts of ecology, things like that. We got real philosophical on it.
Diego, we were kind of thinking we would harass you and make you answer what ecology is, what the scope of ecology is.
I mean, I was just thinking about that when you were saying it. If you want me to, it's totally like, I don't know, all those things, all those different abiotic factors can be wrapped in on it, because it's just a collection of relationships between things that have the capacity to act and have impacts on the relationships like among the whole system. So if it doesn't matter if it's, I think organisms are the best example of it, because they are such good actors. They can cause a lot of, they can take the kinds of actions that result in forming new relationships and maintaining relationships, but in diverse ways. But, you know, the inside of your body is definitely an ecology. The system that involves both a forest and the soil that it grows on, and the continental plate that soil was formed on, and the size of the planet that dictates where that plate is shifting around, and the position of that planet relative to everything else, is all part of the same system. Like, I think that one of the takeaways from this whole thing is that everything is the same system. You can't just, you know, draw a line anywhere between any kinds of definitions and say that that's a real line that marks an actual difference. Like, it's all the same kind of soup mixing around. Maybe, you know, the general definition of ecology has more to do with a system that can perpetuate and maintain the same kinds of interactions that organisms are good at doing. And, you know, in a biocentric way, like thinking about it through the lens of just biological organisms, one that can produce new kinds of bodies, new kinds of actors that can take new actions and generate new relationships that then go on perpetuating themselves.
So it's kind of like thinking about it as this vast system, but rather in the case of, say, Sing, she's peering into a particular moment or happening within this like patchwork, this massive assemblage network that is ecology. And peering into like a specific, like almost breath within that, or like respiratory like flow that appears within that system, as opposed to, so the limitations are open-ended for her. It's not something that's closed, but it's also is something that's particular and momentary. but also shifting and transient.
I think you could probably compare it to in its localized like use to something like capitalism. That, I mean, that's a huge term for a huge process, but that seeks to, and it itself is part of, you know, a more inclusive system, but it seeks to diminish, homogenize the relationships that go on, you know, into subsuming them into property, capital, production of a very narrow set of material and like social or cultural objects that are static, and like eliminating the relationships between organisms, between systems that don't conform to its like set of 3 different, you know, relationships or processes that cause it to continue growing. When you consider it as part of the entire system, you know, you could then, it's an actor in an ecology, but it itself, you might be able to more easily think of it as not an ecology in and of itself, although, you know, that's subject to, that's not a, that's just an impression.
I mean, I think there's a corollary with physics, because you're really looking at what Diego kind of colorfully described as the big soup. And ecology is simply a conceptual tool, a device with which to describe it. And I think that Singh's text elegantly captured the epistemology of it when she referred to the I think it was a Japanese professor who said, well, it just all depends on the type of question you ask, how you describe the answer. So the answer is really the whole soup, but to arrive at any conclusion, you could ask different types of questions and see different relationships. And the trick is not to believe that the one answer that you got the time that you asked that question is the answer. It is an answer. So in a way, the corollary is with the uncertainty principle. The very fact of asking the question already constricts the answer. So you just have to ask infinite numbers of questions and you'll get infinite numbers of answers from the soup. So.
Yeah, so maybe there's a difference between within the soup relationships that propagate or create the conditions for the propagation of different new and different relationships, like, you know, the daimon mating. That's an example of an ecological or a process that lends itself to the creation and maintenance of ecologies or ecological systems where you have different relationships going on, versus systems that try and eliminate the differences and the proliferation of difference in relationships and try and make them static and unchanging and objectified, whether it's classifications, definitions, you know, a wheat field versus a forest, that kind of thing.
Michelle, you said you had so many questions in the chat.
Yes, I mean, I think it's just a matter of level of complexity. Once you reach a satisfying understanding, limited but satisfying, then you kind of, you know, pause it and then move on. But what your pictures were wonderful, and we imagine like, well, he put 2 or 3 color dots, but it's probably with many more colors and many more combination, and we imagine that works probably with many other living systems. So it looks like we are discovering things, and it's not just learning about more complexity, but it's kind of reviewing our whole belief system when it comes to life or species, even within our understanding of what humans are. Looking at other species, where are the connections, where are the overlapping, and that's mind-boggling. So it's very exciting, and I don't think we can do it just tonight, but yes, we have many more questions.
Ooh, sort of one of the questions we did have that was a little more specific was, what are, in the 4 to 10,000 sexes, what are the building blocks, or what are like the, we have our chromosomes that we use to define sex or attempt to, but in mushrooms and fungi, what is it that is, permutated to result in that many categories?
Wow, that's a really good question. Frankly, I am not 100% certain. All I know is that I'm not sure how they determine like what's compatible and not compatible so much to be like, yes, this is a different sex. All I know is that it's like usually just the nuclei within the fungal network that they're picking out, which is what the dots are kind of supposed to represent. Yeah. Also, it's, kind of just, to answer, I don't really know. And that's something I want to know now, even more so. But also with like fungal sex, it's kind of like, in humans, there's like there's just a binary of compatibility within that. But if you're like 10,000, it's just like, well, it's kind of just like, you can just mate with anything that you meet out there in the world. But that's a really good question. I gotta do some research and ask some friends.
Yeah, it would be cool to find out.
I think that reproduction is also situationally dependent, or at least the reading said so. So it's not just a property of the thing, but also a property of the environment in which the thing finds itself. To your sorry. Oh yeah, to your second question about the species and the way that the Chinese demarcated the mushrooms versus the prevalent thinking in the more global scientific research community. I think that is an interesting question in terms of the whole enterprise of science and the peer review process. I mean, to me, one of those that exactly brings up the issue of how science has to be a sort of a consensual human activity, and where the typical sort of tribal and political ramifications of human organization come into play, because the peer review process is highly political, and over and over and over again, we've seen in the history of science how it's sort of disqualified really brilliant ideas at first that have taken generations then later to come back and be proved. So that's a really interesting question that speaks to this idea of, sort of adherence to the group and the proclivity of humans to want to be around others who think the same way they do.
Yeah, I think that's a great point. I think like in the chapter, like the only approach that I can kind of have, like, sorry, the only question I can ask myself is to like reasons to have like this very rigid system and way of understanding is its usefulness. And I don't know that it's, and I think Singh talks about that is that, you know, like, I guess how anthropologists kind of approach methods is how useful is this like very, very strict definition of binomial naming and also DNA sequencing. Like, what does it really, like, what does it really give us in exchange for like where it's come from and also the way it's like shaping our minds to like basically make things invisible, organisms, but also make like, you know, different indigenous knowledges across the world invisible, and delete those. And, you know, there, I don't personally, I don't know if I find it extremely, extremely useful for that purpose. Like there's a way to, good night, Lauren. There's a way to keep track of like biodiversity and stuff like that. But we've had ways of doing that and knowing that for years. It's just like to a much different microscopic level. So in short, I don't know if its usefulness it like lends to its limitations, justifies its limitations.
But its exclusivity appeals to a lot of people because then you can enter the sort of hooded monkdom of people who know how to use the binomial system and be exclusive and special. The Linnaeus system, everyone even admits, I think, in the modern scientific world that it's really hopelessly outdated. I mean, centuries of outdated, but it just remains an international token of exchange in the absence of something that you were referring to that could be a whole sort of different cognitive way of understanding the nature of life and of how we view it. As long as we insist on this sort of molecular level, you know, submolecular level, RNA level isolation of differences, those things that we're going to rely on those kind of devices. But give me a good ethical reason why we need to do that. Or what is the purpose of that? I don't know.
I mean, the whole thing kind of reminds me of like, you know, those pictures, it's like, how many triangles do you see in this picture? And it's just a bunch of triangles, and you could keep infinitely going down and picking out more. And if someone says, you know, a triangle is, there are this many triangles because a triangle is only, you know, above 5 centimeters. Like if you get any smaller than that, it's not a triangle anymore. And they're friends with people who have money and guns. You know, a triangle is going to be a triangle that has that's 5 centimeters long and no smaller, or, you know, it's very simplified, like that's going to be what happens. Even though those categories are not actually, you know, based in a qualitative difference between the groupings that they're categorizing. It's like there was a big shift in evolutionary biology from, you know, using that ranked hierarchical classification where it's like phylum, kingdom, class, order, to just calling things clades and saying like, okay, this is a group of things that are more closely related to each other than anything else. It's like, that to me is a more accurate and compatible definition of groupings and classifications with the system that you're observing. But I guess, I mean, as far as ethical reasons to even make those classifications. The only one that I've heard that really holds any water to me is knowing what to protect and what to prioritize conservation efforts for. And like, if you define this as a group that includes that, but not this, then you can say, okay, well, we need to preserve this patch of relationships. I mean, it's bad. I think it's bad that we even need to be making those triage kind of decisions. I think you should just protect everything wholesale. But I mean, in the worst of scenarios, at least it has that going for it. Not that that justifies, you know, its epistemological history or nature, but you know, that's what I think.
Just the very enterprise, I think, of trying to do that, categorizing things in that way, in a way implies that you know, that we are the gods, really. We have the god's eye view of how to distinguish life, and I think, I mean, I'm sorry, I sometimes skim a lot of science research, but I think I saw something that was where they identified something as matter and antimatter at the same time recently. I can't remember the research, but it was just very, you know, just a quandary. Like, how do you understand that? But in a way, I was like, yes, of course that could happen. Why not? Everything else happens.
Yeah, I think it's a shame that that is the dominant impulse that governs how we relate to these patterns of biological diversity, because, you know, really in the face of this big, I think it was you that said infinity of life, you know, we have the potential to just say, you know, to have our classifications be nothing but, look, this is a guess. This is, I could not possibly make claim on the just overwhelming diversity and heterogeneity and multiplicity of this system. You know, this is just a tiny little line that I've drawn on it that I say, maybe this is how you draw a circle. But, you know,
Are you actually saying that we should be intellectually humble?
Yes, I am. But instead we have people saying, no, here I am as the ruler of this system, which is beneath me and which I have now quartered into little sections, and look at how great I am for being able to, you know, stick a stencil in this pile of dough.
I also think that, like, well, I'm not sure, but I think that the creation, but even more investment in like Linnaean binomial taxonomy also has a lot to do with, well, I'm curious, but don't know, with the need to name things and not rely on local differences of name for the sake, but the purpose of that is for the larger network of extractive industry. Whereas, you know, if you find out, like, rubber trees, right? Like, these people call them that, these people call them that, these people call them that, and you ask, like, well, where is it? And somebody gives you like a totally different name, and you're like, well, I'm just trying to make money and find these trees. Like, so I'm going to tell you that that's no longer that name. It's now this name, so that all of you keep referring to the same thing. So I think also that it's one thought I had about perhaps why it's used so heavily.
Yeah, Ansu, I think that's why Bill Gates would want to, you know, create a blueprint for every species genome.
Yeah, well, absolutely. Yeah, it's a very utilitarian and ultimately nefarious project. And it's the opposite of a stance where you would say life is this incredible potpourri. I'm actually, you know, we're in the midst of it. What we can know is is what we use and what we, just our local place. This is what we know. And our obligation to life is to keep it sort of holistic and not like extract and destroy it. But that would really imply that we have, rather than we dominate and understand and segment and dissect and blueprint everything, that our only obligation to life is to have reverence for it and have an obligation to it. That's a completely different approach, cognitive approach, than let me tell you why this is different from this. And, we've got now even better microscopes to see this, or whatever it is. It's an intellectual exercise rather than a holistic understanding of our role in life.
Yeah, like people taking an entire forest and instead of preserving it and maintaining the relationships that constitute it and letting that persist, cutting the whole thing down so that you can build a museum with enough filing cabinets to put everything inside of and have them stay there in perpetuity with a nice little labeled name. And now the forest is gone, but at least you have a museum with the right name stuck on everything.
I mean, the whole exercise is also a reflection of us as human beings, because what this sort of exercise in finer and finer fine-tuning biology shows is that we have so much privilege put on the one single aspect of our humanness that is placed on a pedestal, which is our rational brains, our ability to look at things in an intellectual sense, versus our humanness as a project of community, including love, aesthetics, spiritual obligation, everything that we need to live.
Yeah, as being participatory members of ecologies rather than consumers and digesters of ecologies.
Exactly.
I mean, ideally, the consuming and the digesting and the rational understanding of an ecology, I mean, could not be mutually exclusive with being a participatory member in the ecology. I think that's what people who have participated in ecologies have shown. I mean, any form of knowledge of a system that you like being a participatory member of an ecology is an extremely generative way of finding out information and classifying and understanding it, like that kind of thing. Hopefully you would not, you don't need to forsake one for the other is what I mean.
Mm-hmm. That's right.
I mean, do we have spores flying in this week's mushroom video?
I don't think we do. Take a look.
Okay, that's all right. Next week, maybe.
Okay.
Ooh.
Oh. Wait, I can't hear Jen. Can anybody else hear Jen?
No.
You're unmuted, but maybe your audio's. I think I know what you're going to say if you just want me to say. I can fill your role, which is.
Can you really not hear me?
Okay, there you go. There you go.
Oh, I was like, I'm unmuted. I don't know what to do after that. I was gonna say, but James probably was correct. I asked if anybody had anything they'd like to share that they were working on. I'm getting closer and closer to finishing my underground microscope. I had a lot of fun this week doing the 2 green tones, which is really a cool shadow effect.
Looks great.
Thank you. I'm having fun.
I don't have my embroidering kit with me today, but so I was just working on another just unrelated drawing, but I guess I can show that. So
yeah.
It's a sea serpent.
Oh, wow.
That's what I got.
God, how
Hey,
talented you
put
were.
that back again for a minute. That's so fabulous.
Extraordinary.
Thank you.
Wait, Diego, did you do some of the drawings for the birds that are in the?
I did all the
embroideries?
birds, yeah.
Okay, okay, I thought so. Well, if nobody else has anything to share, I can send us off with our end of the week question. If that's okay.
Sure.
Cool. All right. So the way Matsutake spores sexually reproduce is beautiful and strange. Fungal mosaics offer open-endedness and a constant flow of new diversity. What inspirations do you take from this encounter? Cool. Well, with that, thanks everybody for the really jam-packed discussion. I know Jen and Cindy and I were worried about this week just because of how technical it got, but that was really fantastic. Thank you, everybody.
Wonderful.
Talking sex never gets too technical.
Thanks, everyone.
Cool.
See you
Bye,
all
guys.
next week.
Bye.
Bye, everyone. Have a nice weekend.
Thanks, everyone.